Good morning. Let's discuss on vitamin K. Vitamin K is a family of structurally similar fat-soluble vitamins found in food and marketed as a dietary supplement as well. The human body requires vitamin K for post-translational modification of certain proteins that are required for blood coagulation. This vitamin is also called as anti-hemorrhagic factor as its deficiency produced uncontrolled hemorrhages due to the defect in blood coagulation. So these are the various families and members of this vitamin K. So one is Dion, that is vitamin K3, which is synthetically produced and it's used as a therapeutic substance for the new neonatal hemorrhagic conditions. Then phylloquinone, it's a naturally occurring found in green leafy vegetables and its structure is in these are all basically naphtha quinone derivative. The side chain is like 20 carbon chain of isoprenoid unit is there. Well, the Mina quinone is also having the same structure. The side chain contains the 30 carbon long chain of the isoprenoid unit. So, this, as I have already told you, vitamin K or phylloquinone derived from the plants, while vitamin K2 or Mina quinone produced by the microorganisms in the intestine itself. Then vitamin K3 or the Mina is a synthetic product. So these are the various members. In 1929, the HDM gave the name coagulation, and it is the Danish word coagulation. So that's how it is known as vitamin K. Then what are the sources? Excellent sources are cabbage, cauliflower, spinach, other green leafy vegetables. The good sources includes the tomato, cheese, dairy products, milk, egg yolk, etc. The vitamin is also synthesized by the microorganism present in our intestinal tract, and the nutritional requirement is 70 to 140 milligrams per day. And biochemical functions of vitamin K. So vitamin K plays an important role in blood coagulation. It is also known as an anti-hemorrhagic factor. Vitamin K is required for activation of clotting factor of factor 2, factor 7, factor 9, and factor 10. These blood clotting factors are synthesized in the liver in the inactive form and are converted to the active form by post-translational modification of the gamma carboxylation of glutamic acid residues. And in this, the vitamin K dependent carboxylase enzyme adds the extra carboxyl group at the gamma carbon of the glutamic acid residue of inactive blood clotting factor. So we'll see in the next slide how the activation happens. So this is the basically conversion happens in the glutamic acid residues of the clotting protein. So this is the component of clotting protein, this glutamic acid, and vitamin K dependent carboxylase, which is abundantly present in the liver. In the presence of carbon dioxide and oxygen, it converts it into the gamma carboxyglutamate in the protein. So it was initially only one carboxylic acid group in the side chain. Now it is there are two carboxylate groups are present. So and these groups can abundantly chelated or winds up with calcium. And in this stage, the prothrombin can easily attaches itself to the platelets, and this is how it is being activated. So in the liver, as we know, the vitamin K dependent carboxylase is present in the liver. So the cycle happens in the liver. This is the reduced form of vitamin K, that is the hydroquinone form. And there is an in course of conversion of glutamic acid to gamma carboxyglutamate in the presence of carboxylase enzyme, it converts up into the epoxide form. So this is the epoxide form of vitamin K. Then further epoxide reductase enzyme, it converts further into or reduce it into the quinone form. So this is the oxidized form of vitamin K. And in course of this conversion, there is a reduced lipoic acid converted up into the oxidized lipoic acid. Then further, this oxidized vitamin K or quinoid form of this vitamin K again revert back to the hydroquinone form of vitamin K in the presence of vitamin K reductase enzyme. So this is how the cycle goes on in the liver. And this site um, which is being mediated by epoxide reductase can be inhibited by the two important inhibitors, dicumarol and warfarins. So these are the competitive inhibitors of vitamin K. They inhibit the action of vitamin K. So this is the structure of dicumarol. So this is competitive inhibitor. Structure is more like that of vitamin K. So that's how it is in competitive inhibitor. And same is with the warfarin. Then deficiency manifestations. The vitamin K is also produced by the intestinal microflora. Has the dietary deficiency rarely occurs or normally doesn't occur. The vitamin K deficiency is associated with hemorrhagic diseases. Uncontrolled hemorrhage occurs on minor injuries. So in vitamin K deficiency, the bleeding time and the clotting time of the blood is increased. So vitamin K deficiency, however, is found in a few cases like in biliary obstruction. So biliary obstruction inhibits the entry of bile salts to the intestine because the absorption of vitamin K happens with chylomicrons and bile salts. So that's how there is a deficiency in absence of or in conditions of biliary obstruction. In the newborn infant, because their placenta is not able to pass the vitamin to the fetus efficiently, and the gut is also immediately sterile after the birth. No microfloras are there, so that it cannot have its own synthesis of vitamin K. So they may also suffer this kind of deficiency. Then following prolonged antibiotic therapy, the get also sterilized. So overexposure of antibiotic lead to killing of the bacteria or the gut microflora. Then also in fat malabsorption. So these are the few conditions which lead to the deficiency of vitamin K. So hypervitaminosis K. So excessive doses of vitamin K produce a hemolytic anemia due to the increased breakdown of RBCs and jaundice or kernicterus, which happens in the newborn babies. So this deficiency leads to the increased amount or concentration of bilirubin, which can damage the brain also. So these are the hypervitaminosis effects of vitamin K. Thank you.